So as you know, the blood vessel barrier impedes the drug delivery. How to optimize it? We've tried, it has been tried EV high-dose chemotherapy, as you know, intraarterial. There is also convection-enhanced delivery. A good team is doing this in Bristol, in England. Thanks to INN, we know that now that the microbubbles and ultrasound can open the blood-brain barrier. And my key concern as being a neurosurgeon is how we can make this every month for every chemo session, 10 times, 12 times on a patient. And as a neurosurgeon, I'm accustomed to do a bone flap and to do burr holes, so I would say, why not using those burr holes to put transducers in it? So this is a start of the story. And the device is implanted inside the bone, so there is no bone between the device and the brain. And it is transdermally connected to a generator in order not to have any problem of MR compatibility afterwards. So it's a 10 millimeter transducer, 1 megahertz. It's implanted during a regular surgery, and you have a transdermal needle to give the power. We did a lot of preclinical study that I have presented already here on primates, on baboon and dogs, and all the different types with pet studies. All this is published now. We have some survival, progression-free uptake with carboplatin on rat models. And here is when the patient comes. Actually, the system is implanted, and you have a 2 minutes sonication. So you have to plug the device in, the needle in, and you give microbubbles, and you have the 2 minute sonication. And afterwards, you have a 6 hours opening. It's one center trial, 5 escalating dose, with a primary endpoint of safety. You see this patient has a left opercular blood-brain barrier opening inside the language area, and there is absolutely no problem of function. She can totally speak correctly. This is all the patient table. So we did by now 41 sonication. And we set up a table in order to see the type of blood-brain barrier opening. I don't know if it is correct for the future to use, but we've noticed that the first step is to have subarachnoid contrast enhancement. The second step is have to gray matter enhancement. And finally, if you increase the pressure, you have the white contrast enhancement. So you see that there is some white contrast enhancement at 1.1 megapascal, so it should be maybe the correct pressure for the future. Here is an example of a patient. You see pre-sonication and right after sonication, the MR right after. You see very well the contrast enhancement in the field of the ultrasound transducer. We have shown in previous studies that the carboplatin is increased by 5 in this area, but also the carboplatin diffuse all around for about 7 to 10 millimeters. So the field of action is larger than what you see on T1 contrast enhancement. This is a coronal view. You see the device that is totally MR compatible, that's just like a dot. And this is a patient 15, the same one actually, who actually has a pretty nice tumor control. These all are recurrent glioblastoma, so they expect to have recurrence at 2 to 3 months after carboplatin injection. This patient is at the 10th sessions without any recurrence. So we're very proud of this patient. We have other patients that have gone or their 5th session, 5th chemotherapy, so 5 months progression-free survival. Another interesting point is to see that over time, in some patients, we have some flare clearance. I mean, some flare less hyper single flare images. meaning that we're not that we are inducing some benefit in the infiltrated brain. So for 15 patients, 41 ultrasound treatment, no hemorrhage, no fluorodema, no dose-limiting toxicity by now, and it's pretty rapid. It takes mean time is 8 minutes, and we will go up to 19 patients in the future. Thank you very much.
Thank you, that's great. Do we have any quick questions from the audience? How long do you anticipate you're going to be following these patients over?
I am following up until they die. Yeah.
Okay.